back on the journey of microbiology. We started discussing the chapter General Characteristics of Microbes. In this chapter, we studied about the bacterial structure, how bacteria morphologically, how they are appearing. And in this particular lecture, we will study about the shape of bacteria along with the adaptations which they adapt during the unfavorable situations, the unfavorable conditions. Then we will also study about the colonizations. Okay, so hope you all are clear with the old concepts. Now see the slides, the lecture outline. We already covered about the structure and classification of microbes. We studied the microbiology, the morphological types, size and form of bacteria, the mortality, the movement capability of bacteria. Today in this particular lecture, we will study the colonizations, growth and nutrition of microbes. Okay. So the first topic is about the shape of bacteria. The first is the shape of bacteria. There are four shapes. There are four shapes of bacteria generally. And these shapes, the first shape is round shape. The round shape bacteria called the coccus. Coccus shape is a round shape bacteria. Then we have another type called bacilli, which is a rod shaped. Then we have another type spirula, which is a spirulum size, and then we have comma size vibrio. So we have basically the four size of bacteria. The four on the basis of shape. On the basis of shape, bacteria are categorized into the four groups. Bacteria are categorized into the four groups. The first group is coccus. The first group is coccus. The another group is vibrio. The another group is vibrio. Then the third type is spirulum, and the fourth type of bacteria is the bacilli. The bacilli. So on the basis of shape, bacteria is categorized into the coccus, vibrio, spirulum and bacilli. Okay, coccus is a round shape bacteria. Coccus is a round shape bacteria. It's a round shape. Okay, then vibrio is looking like a comma shape. It's a comma shape. Commas, like this, round comma shape. There's spirulum and bacilli. Spirulum is basically the flagellar type. It's a flagellar type. Okay, like the flagellar type. Then we have bacilli. Bacilli is a rod shape. Okay, so on the basis of size, bacteria is categorized into four groups. The first one is called the coccus, which is a round group. Second is vibrio, shaped like a comma. Then we have spirulum, shaped like a flagella. And then we have bacilli, shaped like a rod. Okay. In previous video, we already discussed about the gram-positive and gram-negative bacteria, basically differentiated on the basis of gram reagent, and their cell wall is also different. Okay, so sometimes in the textbooks, in the other books, you find that something called round gram-negative bacteria. Okay, you find also the bacilli, rod gram-positive bacilli. Okay, so these names appear just because of their names on the basis of their shape. Okay, so now let's see the slides. Hope you clear with these all the shapes of bacteria. Now we have spore and sporulation. Okay, so first I will describe it and then I read the slides. Okay, so we have two things. The first is spore and sporulation. See, what happened? During the harsh condition, environmental unfavorable condition, bacteria adopt certain kind of modifications that help bacteria to flourish in those extreme conditions. Okay, so on that period of the time, their growth and metabolism is formed in the form of spores. Okay, and the formation of spores during the unfavorable conditions, the phenomenon is called as sporulation. Okay, now for example, if this is one typical bacterial structure, we will see in our previous lectures that bacteria is covered by one covering, first covering, that is a cell wall. That's a cell wall. Then we studied bacteria sometimes also covered by one more layer, that's called the glycolex, slime layers. Okay, glycolex or a slime layer. It's a glycolex or slime layer. Now that's one typical bacteria. Okay, it won't be live bacteria until unless it don't carry the genetic material. So in the case of bacteria, the genetic material is not organized, we already discussed. Okay, and that genetic material is called the nucleotide. Apart from the nucleotide, they have other cytoplasmic structure as well. The first structure is called the ribosomes. It is widely dispersed in the cytoplasmic space. Along with these ribosomes, they carry also the mitochondria, a powerhouse double membrane bound organ of the cell. They have multiple these mitochondria. Then for motility purpose, they carries the pili. They have in the outer surface which invegetate till the cell wall. Okay, they have this structure called the pili. Then we studied they carry, some of the bacteria carry the flagella also. Okay, flagella is a short, it's a thicker and short. The main difference between the flagella and it is present 1 or 2 in number, and the pili is present more in the number. Okay, they help in the adhesion and attachment, capturing the food, phagocytosis probability. All this is already we discussed. Now today our point is that's a normal bacterial growth. Okay, if I tell you the normal bacterial growth, in normal conditions, forget about this extreme condition. In normal condition, bacteria does what? It undergoes one type of asexual reproductions. We call it binary fission. In normal condition, I am talking about the normal condition. In normal conditions, they undergo one type of reproductions that's called the binary fission. Okay, that's called the binary fission. As the name suggests, binary means dual, double. So it will divide into two small bacteria. I am roughly just describing it. Is divided into two small bacteria. That's the genetic material. Okay, along with all the structure are also present. That's a normal bacterial growth. That's a normal bacterial reproduction. They take in the nutrients from the outside, they grow their size, their nucleus undergoes the cell division, and they divide into the two groups and this form. That type of we can say the division is called the binary fission. Okay, it shows in the favorable condition when the nutrients are abundant, the temperature, we can say the light, okay, the amount of air, everything is adequate, then bacteria perform the binary fission. But what happened when during the burst of, we can say, the extreme conditions, sometimes bacteria won't get nutrient, sometimes bacteria get attacked by the, we can say, the antibiotic therapy, antimicrobial therapy. So on that situations, bacteria does what? It start one process that's called the sporulations, a formation of spore. It's happened during the unfavorable condition. Remember, it's happened in the unfavorable conditions. Okay, unfavorable condition. Now this bacteria grow, okay, they grow, and after they growing, their nucleus, their this genetic material, not a nucleus, it's a nucleotide, because they are not enveloped, they are a prokaryotic cell. So what happened, these nucleotides undergo the division, let me show you. It undergoes a division, okay, into two forms, and then this part they spelled out, that's called the invagination, okay, that's called the in out. And now what happens, see, one nucleus is within intact with the original bacterium, and another will appear on their surface, see, okay, they propel out, then they engulf this, that's a stage, and then again they come to the same state, okay. Now they have the two genetic material, see, and they form the covering of the second copy of their genetic material, they covered. This process is called the spore formation, okay, spore formation. Now what are the clinical importance of newly formed spores? See, in bacteria, basically the gram-negative rods, they undergo this sporulation process. What happened about the clinical significance of this spore? The clinical significance of spores is they are multi-layered. It is multi-layered, first of all. Second thing is it is highly resistant to the antibiotic. First, the clinical significance, if we seeing, the first clinical significance associated with these spores are they are multi-layered. They won't want too much nutrient for their survival, okay. If you're not given the water, if you're not given the nutrient, for sometimes they won't feel any fluctuation in their shape, their metabolic process, okay. So they won't need too much, we can say, the too much nutrient, okay. Then they are highly resistive, they are highly resistant towards the antimicrobial therapy, we can say the antibiotic, they are resistant towards the antibiotics. Clear? That's a three fundamental, we can say, the clinical significance related with the newly formed spore, okay. And then when the favorable situations came, these highly resistant cell wall formed spores need to be dissolved. So bacteria secrete certain kind of enzyme during the favorable situations, and then what happened, the covering of the spores will rupture and the progeny of bacteria will come out, and they will again divide it in the form of binary fission, okay. So about the spore and spore formation, you always keep in the mind, it's always happen during the unfavorable condition, first of all. Second thing is, it is a mode of, we can say, the replication, it's the mode of the reproduction in the bacteria during the unfavorable conditions. What happened, the genetic material of the bacteria, that's a nucleotide, they divided into the two parts, they expelled out, then they invegetate those, phagocytosed those parts, again encropped into this and formed one multi-layered cyst, we can say the cyst formation, the wall formation they formed, and this is highly resistant towards the antibiotic. They don't need nutrition and they are multi-layered, okay. Now see the slide content, the spore and the sporulation. In response to the challenging environment conditions like limited nutrients, certain gram-positive rods employ a remarkable survival strategy. It's a remarkable survival strategy for the bacteria. These bacteria undergo extensive structural and metabolic transformation, culminating in the development of a dormant cell known as endospore, okay. Now these spores, they are known as endospores because they are formed inside the bacteria, so they are known as the endospore. These endospores helps them to survive tough conditions until things get better. Okay, it's like their own tiny survival capsules. I already told you, no, that's a multi-layered, resistant to antibiotic, as well as they won't need. Example of gram-positive bacteria is Cholesterum tetani, causing the tetanus. Then we have Corynebacterium diphtheria, causing the diphtheria. Then we have Clostridium difficile. It's a antibiotic resistant, antibiotic associated diarrhea, okay, and Bacillus anthracis. These all kind of gram positive rot bacteria, they undergo during the unfavorable situation the sporulations. Now see in the another slide, they are remarkably resistant to heat. I already told you, no, they are resistant to the heat and antibiotics. Okay, desiccations, ultraviolet light, and bactericidal chemical agent. Okay, bactericidal chemical agent. Now see the word, there is a two term, you always come across these two terms. Let me define it. The first term is called, okay, now you are clear with this. The first term is called the bactericidal. The first term is called the bactericidal. Okay, bactericidal. And one parallel word, the meaning is different, is called the bacteriostatics. Bacteriostatic. See, these are basically the two kind of, we can say, the chemical compound. In the bactericidal chemical compound, like for example bactericidal antibiotics. See, bactericidal antibiotics, these bactericidal antibiotics, they kill the bacteria, whereas the bacteriostatic kind of antibiotics, they basically suppress the growth of bacteria. They suppress the growth. Okay, so cidal means death and static means suppress. Okay, now see the slide. In fact, sterilization procedure are assayed by their ability to kill spores. Sporulations can be thought of as repackaging a copy of bacterial DNA into the new form that contains very little water. It has greatly reduced metabolic activity, does not divide, and has a restructured, highly impermeable multi-layer envelope. The multi-layer covering I already told. Now the spore germination. Spore germination is basically the formation of a spore in the favorable conditions. When the favorable conditions come back, the spores is converted into the normals, we can say the bacterial genetic material, the bacterial progeny. Then the medical significance of sporulations. I already told you the medical significance of sporulation is, the first is we have to see which bacteria is resistant toward which antibiotics. Okay, so during the unfavorable situations we cannot bombard with the antibiotic to calm their growth. Okay, so we have to understand what is the meaning of sporulations as far as the medical importance is concerned. Okay, now see the one diagram I already draw in the board. This diagram shows how spores is formed in the bacteria. See what happened, the plasma membranes undergo the division, okay, and their genetic material is shifted into the two parts. Then what happened, those genetic materials, one genetic material is like the parental, their own main genetic material, which is persist with the normal location in the cytoplasm, and the another you seeing this genetic material, they are separated. Okay, they are invaginated, matlab expelled out, and this first formed expelled out nucleotide is called the 4-spore. It's called the 4-spore. Then what happened, bacteria engulfed those four spores. Bacteria engulfed those four spores and then formed one thick layer around the four spore. And this thick layer formed layer on these four spores and transform into the endospore. And then during the favorable conditions, they release out. These endospores are released out in the form of normal progeny. Now, growth and metabolism of bacteria. See, growth is basically, we can say, the increase in the body mass and as well as the cell number. Growth is characterized by the two things. The first thing is increase in the body mass, and second thing is increase in the cell number, So in bacteria, they also undergo the growth. In order to leave their progeny in another generations, for the another generations. Okay, so for that they undergo the two things. The first one is metabolism. And the second thing is reproduction. First thing is metabolism. Like bacteria first need the nutritions to grow their cells, okay, and then they will leave the progeny by the means of reproduction. And which kind of reproduction? Yes, binary fission, the normal division, 1 divided into 2, 2 divided into the 4, 4 divided into the 8, the arithmetic division. Okay, the arithmetic division, that's called the binary fission. Now see the slide. Bacteria metabolism involves the conversion of nutrients into energy with variations like aerobic and anaerobic pathway. Let me define these two words, aerobic and anaerobic. See, aerobic, as you know, that aerobic is basically in the presence of oxygen. Okay, so some of bacteria have the capability to perform their metabolism on the presence of oxygen, whereas some of the bacteria have capability which form their metabolism during the absence of oxygen. So the category which need oxygen, that's called the aerobic bacteria, and the category which don't need oxygen, that's called the anaerobic bacteria. Now, growth includes binary fission, where a single bacterium divided into the two identical cells, allowing population to increase rapidly under the favorable condition. Let me tell you one thing. Identical twins, identical copies. Bacteria don't show the variation. As far as the human is concerned, we are a developed organism. Okay, whatever color of father, mothers we had, okay, we don't carry the same color. We have certain variations. Okay, so sexual reproduction shows the variation. In order to color, character, okay, color, intelligence, everything differ from father to mother, from one generation to the next generation. Nothing is like persistent. Okay, but in the case of asexual reproductions, what happened? Whatever shape, whatever the normal machinery is present, the normal genes is present in the main progenitor cell, like the main bacteria, same thing is directly copied into the next generation. They won't show the variation. That's the fundamental difference between the sexual reproduction and asexual reproduction. In the case of sexual reproductions, they preserve the variations, whereas in the case of sexual reproductions, they shows the variation, the phenomenon of variations. Okay, and in the case of bacteria, that's asexual reproduction, binary reproduction. In this case, bacteria preserve their character throughout the life cycle. Let me show on you the board what I described. Okay, it's important to understand. It's important, and also the understanding is like it's interested to understand. See, the normal human, for example us, it's like a human, we undergo this type of reproduction that's called the sexual reproduction. Okay, sexual reproduction. Whereas the prokaryotic organism, or we take the example now, the bacteria. Okay, bacteria, during the normal favorable conditions, they undergo the asexual reproduction. reproduction. See, sexual reproductions emit, they show the variation, they show the variation. Okay, now asexual reproduction, they preserve their identity, means they preserves identity. It means what? Whatever characters, whatever the type of genetic material is present in the bacteria, same thing is transferred into their progeny, the newly formed bacteria. But in the case of human, we show the variation. We are different from our parents in so many contexts. As a color wise, as a intelligence wise, as a height wise, all the character wise, we differ from our parents. Okay, so and why this happen? Why they show the variation and another not show the variation? That's a basic cytology. You know, during the division, cell division, the human cells, or we can the germ cells, they show the crossing over. They show the crossing over. But in the case of asexual reproductions, they won't show the crossing over. So crossing over leads to the variation. The chromosomal crossing lead to the variations, whereas there is no any chromosomal variation. This is a primitive type, old type of reproduction, asexual reproductions. So their identical twins, they produce the identical twins. Clear? It means bacteria won't die, they are immortal. Their just genetic material is divided. Means I am a bacteria, okay? Now I divide it into the two parts. I am dying? No. I am living in the two forms. One from here and one from here, okay? So generally, technically, we can say the bacteria are immortal. Now, see the slide. Metabolic characteristics, okay? Human pathogenic bacteria are chemoorganotrophic, driving energy from the breakdown of organic nutrients, okay? They taking the energy basically from the dead organisms, okay? They oxide nutrient substrate through either respiration and fermentation. Now see the slide. The metabolic process which is occurring in the bacterial cell, metabolic process in the bacteria get into two things. They performed in the two bases. The first is called the respiration, and another one is fermentation. For energy, for synthesizing the energy, they undergo the two process, okay? First is respiration, second is fermentation. In the respirations, they need oxygen along with the adequate temperature or susceptible temperature, we can say, okay? And in the case of fermentations, they don't need oxygen. So it's clear that anaerobic bacteria shows the phenomenon of fermentations, whereas the aerobic bacteria shows the process of respiration. Means aerobic bacteria derive their energy from the respiratory phenomenon, whereas the anaerobic bacteria, they derive energy from the fermentation activity. Now see the slide. Respiration and fermentation, they oxidize nutrient substrate through either respiration or fermentation. Respiration involves oxygen as the electron and proton acceptor, while fermentation uses organic molecules to this purpose, okay? Classification based on the oxygen requirement, I already told. Some of bacteria need oxygens, they are called the aerobic bacteria. Some of need the without oxygen, they are called the anaerobic bacteria. Then cultivation method. See the slide. Cultivation method is basically forming the bacteria, okay, growing the bacteria on the nutrient plate, the cultivation. For cultivation, we need first identification of bacteria. We need a proper identifications. In this successive lecture, you will come to know that all bacteria cannot grow in all kind of nutrient media. Okay, they have their selected media type on which only the specific kind of bacteria can grow. Clear? So first we need to the identification. Second, we have to set a proper, we can say the proper environment, we can say the necessary environment, or in another word we can say the adequate environment. Okay, adequate environment. And another thing is we have to wait during certain period. There is a one life, we can say life, or leave it the life, that's called the growing time. Clear? These three things is needed in cultivation. These three things is needed for the cultivation of the microbes. Clear? Now see the slide. Nutrient both on the agar containing agarose is used for bacterial cultivation. Okay, nutrient agar liquefy at the 100 degrees Celsius gels and the 45 degrees Celsius. That's the standard temperature. I already told you, adequate environment. They won't grow in each and every temperature. They need a proper scale of temperature for their growing, their cultivation. Now the diagnostic bacteriology. Bacteriology is basically, we are suspected, okay, this person is maybe their sign and symptom shows that okay, this kind of bacteria may be present in that. Then we taken the sample and send it to the lab. This is called the diagnostic bacteriology. So selective and indicator medium play a crucial role in diagnostic and bacteriology. We already discussed about the reproduction and the generation time. This growing time is also called the generation time, and in the case of bacteria, it takes 15 to 30 minutes for proper longer growth. Now growth curve phase. See the slide, see the board. Growth curve phase. Microbiologists are able to present bacterial growth on one graph. That's called the bacterial kinetic graph. In bacterial kinetic graph, they simply drawn one graph. Okay, in one scale they taken the time. Okay, and in the next scale they taken the number of cell. Okay, the number of cell. Okay, and they mark the scale. Now what happened? Bacteria initially, that's a 0 point. Okay, bacteria, change the marker first. Okay, bacteria first shows one stationary phase, then they make one growth. See, that's a bacterial kinetic curve. Bacterial kinetic curve. It's very important to understand. Bacteria shows this kind of growth curve. Okay, you are, as see the slide, in the slide you are seeing the first phase is called the lag phase. This phase is called the lag phase. Okay, then in the lag phase they first grow slowly, and then they make too much growth. Then they make the too much growth, and after that you seeing this curve become the straight. their growth is suspended, and this point is called the stationary phase. Stationary phase. Okay, and then afterward, see the curve is declining, is deteriorating, and this decline curve denotes bacteria is dying. So it's a die point. Okay, this is a one typical bacterial kinetic curve. Kinetic means speed. It means speed. So it's basically the speed which is showing bacterial growth. Okay, which is showing bacterial growth. Initially, they take in the nutrients, they grow slowly, then they make one, we can say, the large spike, the large growth, then their growth is suspended here, they won't grow from here, okay, and then slowly they will die over a period of time. Now see the slide, how beautifully it's represented. In the lag phase, I already told. Then one phase I forget to mention, that's called the log or exponential phase. Log or exponential phase. Log, this phase, or exponential phase. Now see, there is a bacterium, one bacterium, singular bacteria is called bacterium. One bacterium they take in the nutrients, okay, and then they show a proper pattern of growth. They show a proper pattern of growth, okay, and scientists, microbiologists are able to interpret those growth in the form of graph. So this is basically the bacterial kinetic curve, bacterial growth curve, okay. In initial period, they grow slowly, okay, that's called the lag phase. That's called the lag phase. They are not metabolically too much active. Then they take in one long growing period, that's called the stationary phase. That's called the stationary phase. Then that's called the log or exponential phase. Exponential always means in the form of power. It's like too much multiplications, okay. Exponential phase. Then their growth become suspended, okay, and they slowly they detorted, their body start detorted, declining, that's called the death phase, okay. Now see in the slide, it's beautifully represented, okay. Then we already described about the overview of respiration, fermentation and energy in productions. Now see only the slide diagram and you can able to see it. I am not giving you the too much descriptions because you already studied this in the biochemistry, that in the fermentations they undergo the pyruvate breakdowns and they extract energy from there. Glucose undergo, in the absence of oxygen, form the pyruvate, and from the pyruvate the molecules of electrons is released and they get energy. In the case of respiration, they undergo the process of called the glycolysis. Though is beautifully presented, the fermentations and the aerobic respiration. Now come to the point colonization. The word colonization, it is the last topic for today. It's the last topic for today, colonization. Colonization is basically the formation of bacteria on the surface. It might be the human body, any object, okay, favorable object where bacterial growth can sustain, they can flourish, as well as in the animal's body, So this colonization, see the slide, bacteria can colonize various environment including the human body, soil and water. Factors like nutrient availability, temperature and pH influences their colonization pattern. In some cases, bacteria form biofilms, complex community attached to the surfaces, enhancing their survival and resistance to antibiotics. Understanding the bacterial colonization is crucial for areas such as medicine, agriculture and environmental science. So it's nothing, it's just a formation. It's a formation of bacteria on the different places, okay. So for the growth of bacteria on the different places, they need a proper adequate environment, okay, the proper temperature, light, humidity, okay, everything is required for their normal formation of colony, formation of their, we can basically say the ecosystem of bacteria. Okay, now see one diagram, the growth of the bacterial colonies on the solid media plate. See, this is one agar plate, and that agar plate we put in the single bacterial cells. Okay, and over a period of time their number will increasing, okay. In the 0 hour, the number of cell is 1. In the 4 hour, the number of cell is 256. See how fastly growing they are. 256. In the 8 hour, they become the 65,000. Okay, and in the 12 hour, they become the 17 crore. That's the visual colony. 70 lakh, sorry, they become the 70 lakh, that's the visual colony. Okay, let me draw it here so that you can visualize it clearly. Okay, sorry. See, we taken one agar plate. Okay, we taken the one agar plate. Agar plate basically the media. Okay, it's a which kind of media? It's a solid media. Okay, it's a solid media. So we taken the agar plate, and the bacteria which can grow here, I give you the name, that's called the E. coli. It's present in our intestine. Okay, it's present in our intestine, and when they're abnormal, they are present in a normal flow. I already told you, no, there are some bacteria which is living inside very peacefully in a commensal level. We and bacteria eaten together in our body. Okay, our cells and bacterial cells also eating together, until unless their number won't get high. Okay, if their number will get high, they causes certain signs and symptoms. So leave that point. Normally in the normal flora, E. coli live in our body. For the E. coli growth, the type of media plate is needed is called the agar plate. Okay, agar media. They need the agar media. So we put one cell of bacterium in the agar plate. Okay, then and here we interpreting two things. Okay, time versus growth. Time versus, we can say the cell number or growth. Okay, cell number. During the 0 hour, it's the starting period, they won't show any growth, 1 in number. Okay, then in 8 hour, they become how much? Sorry, in the 4 hour, they become 256. Okay, in the 8 hour, 4 hour, in 8 hour, it become how much? It becomes 65,000. It's become the 65,000. Okay, and in the 12 hour, it's become 1,70,000. It's become 1 crore 70 lakh. In 12 hour, it become the 1 crore, okay, 70 lakh. In the 12 hour, bacterial growth time is too fast. It needed too fast growth. Okay, so during this much growth, we can see bacterial growth on agar plate. Normally, we cannot see bacteria without microscope, but when we grow this on the bacterial plate, the agarose plate, the solid media, and then they form one mold-like structure, one mold-like growth, and that's visible by the naked eye. So for concluding this chapter, today we studied about the extreme condition. In the extreme conditions, how bacteria flourish their life. In that we studied about the spore and the sporulations. Okay, after that we studied the normal metabolic process of bacteria, how their growth happened. We studied their respiration, fermentations, along with their reproduction, the binary fission. Okay, then at last we studied about the growth bacterial curve, growth kinetic curve. Then we studied the normal growth on the culture plate of bacteria, taking the example of the Escherichia coli. So thank you with having me. Bye bye. Hope you understand this lecture. Bye.